Antibiotic resistance is a global health threat. In 2019 alone, an estimated 1.3 million deaths worldwide were attributed to antibiotic-resistant bacterial infections. Wanting to contribute to a solution to this growing problem, researchers at Baylor College of Medicine are examining the process that drives antibiotic resistance at the molecular level. In journal Molecular Cell, they report the crucial and surprising first steps in increasing resistance to one of most commonly prescribed antibiotics, ciprofloxacin, or Cipro for short. The findings point to potential strategies that could prevent bacteria from developing resistance and increase the effectiveness of new and old antibiotics. Author Susan M. Rosenberg is the Ben F. Love Chair of Cancer Research, Professor of Human Genetics, Biochemistry, Molecular Biology, Virology, Microbiology at Baylor. She is also the program leader at Baylor's Dan L. Duncan Comprehensive Cancer Center. We discovered that Cipro triggers cellular stress responses that promote mutations. This phenomenon, known as stress-induced mutagenesis, produces mutant bacteria, some of which are resistant to Cipro. Cipro-resistant mutants continue to grow, maintaining an infection with Cipro that can no longer be eradicated. Cipro causes breaks in DNA molecule that builds up inside the bacteria and ultimately triggers a DNA damage response to repair breaks. The Rosenberg lab's discoveries regarding steps involved in stress-induced mutagenesis revealed that two stress responses are required: the general stress response and DNA damage response. Some of downstream steps of the process leading to increased mutagenesis had previously been uncovered by Rosenberg lab and colleagues. In this study, researchers discovered molecular mechanisms of first steps between antibiotic that causes DNA breaks and bacteria that initiate DNA damage response. First author, postdoctoral assistant in the Rosenberg lab, Dr. We were surprised to find an unexpected molecule involved in modulating DNA repair, said Yin Zhai. Usually, cells regulate their activity by producing specific proteins that mediate the desired function. But in this case, the first step in activating the DNA repair response was not about activating specific genes that produce specific proteins. Instead, the first step consisted of disrupting the activity of RNA polymerase, an already existing protein. RNA polymerase is the key to protein synthesis. This enzyme binds to DNA and transcribes DNA-encoded instructions into an RNA sequence that is then translated into a protein. We discovered that RNA polymerase also plays an important role in regulating DNA repair, said Zhai. A small molecule called nucleotide PPGPP, found in bacteria exposed to stressful environment, binds to RNA polymerase via two separate sites required turn on the repair response and overall stress response. Interfering with one of these sites shuts down DNA repair, particularly in DNA sequences occupied by RNA polymerase. Professor of Molecular Human Genetics, Molecular Virology Microbiology, DLDCCCC member co-author Dr. PPGPP tells it stop-go back to repair the DNA by binding the DNbound RNA polymerase, said Christophe Herman. The Herman lab found the repair-RNpolymerase link previously reported in Nature. Rosenberg's lab discovered that DNA repair can be an error-prone process. As repair of broken DNA strands progresses, errors occur that alter the original DNA sequence, producing mutations. Some of these mutations will confer bacterial resistance to Cipro. Interestingly, the mutations also trigger resistance to two other antibiotic drugs that bacteria haven't seen before, said Zhai. We are excited by these findings, said Rosenberg. They open up new opportunities for designing strategies that will interfere with the development of antibiotic resistance and help change the course of this global health threat. Moreover, Cipro breaks bacterial DNA in the same way that the anti-cancer drug etoposide breaks human DNA in tumors. It also helps to combat cancer chemotherapy resistance. We hope it can lead to new tools. Other contributors to this study include P.J. All of the Baylor College of Medicine, Hastings, This work was supported by National Institutes of Health grants, R35-GM122598 R01-CA250905, NI Directors Pioneer Awards DP1-AI52073, DP1-AG072751 and DP1-CA174424. Cell Sequencing Core with BCM Cytometry CPRIT Core Facility Support Award. CPRIT-RP180672 and NI Awards CA125123 and RR024574.